We'll give it one more minute and then we'll get started.
I'll give people a warning before I start. I'm maybe on the verge of losing my voice. I'm hoping it holds out the whole time. I don't have to tag out for Alyssa, but I think I can make it through. But if I'm taking a lot of sips of water, that's why.
Oh no.
Luckily, it's not COVID or anything. I was just yelling too much at a sporting event, so that's my own fault.
Okay, well, Melissa, if you're ready to go, we can get started. It's a couple minutes past the 12 o'clock hour. So thanks to everybody for coming today. And I'd like to introduce Melissa May with many of you already know her from the San Juan Soil and Water Conservation District in New Mexico. And she's going to present today about the microbial source tracking project they did in the San Juan watershed. So take it away, Melissa. Great to have you here.
Yeah, thanks, Christine. It's good to be here. And thank you all for coming today. I've been involved in this project, and some of you have probably heard me talking about it for the past 10 years. It kind of had its initial conception back in 2012 and our big original sampling in 2013 and 2014. But I do see some new names here, and so hopefully some of this will be new to all of you. And the most exciting part is presenting our new data from 2021. But first of all, I do have to give a huge shout out to Alyssa Richmond. She's our San Juan Watershed Group coordinator. She put the majority of these slides together and did pretty much all the analysis on the new bacteria data. And so huge shout out to Alyssa. I may rely on her for helping with some of the question answering and that type of thing. And she will be giving a really similar presentation this week at the New Mexico Weary Animas and San Juan Watersheds Conference. So if any of you are attending that, you'll get to hear Alyssa's version on Wednesday. I also want to start by thanking our funders. All of our recent data collection was funded with an EPA Clean Water Act 604B grant. And so Heidi Henderson is here. She's our grant manager for that. So thanks, Heidi. We are really lucky to get those funds and then still have original matching funds from our original BHP Billiton study. And so that's what funded this work. And I will go ahead and get started. So most of you are probably familiar with the San Juan Watershed Group, but basically the Watershed Group is a local stakeholders group of citizens and agencies working to improve water quality within the San Juan River watershed, and really looking at collaborative solutions to protect and restore water quality. So not regulatory, really focusing on research, planning, on the ground, non-point source pollution control projects, and community outreach and education, which is why I'm here today. So looking at the water quality impairments in the New Mexico portion of the San Juan watershed, that's mainly where we're focused. And bacteria has been the major focus of attention for the San Juan Watershed Group since all of our local rivers have been impaired by E. coli at one time or another. So if you see here on the map in purple, this was the 2016 to 2018 impairment listings, and you see these purple lines. All of those stretches were impaired for E. coli back then. Some of these, the 2016 listing, a lot of this data was based on some of our 2013 and 2014 data collection. We had up to 50 samples per site during that time period, so really high density of samples that was submitted to the state to go into their impairments listing. If you keep an eye on those purple lines, you can see how that has changed. The 2020 listings, NMED had come up and done additional sampling in 2017 and 2018 that did not find as high concentrations of E. coli in several reaches of the river. And so going back and forth, you can see the purple impairments disappear off of the Animas River and off the upper reach of the San Juan River. But the La Plata and then downstream of Farmington, the Hogback was still impaired for E. coli, as well as some downstream tributaries. This is Stevens Arroyo, Shumway Arroyo, and then Gallegos Canyon. Those were all added to the impairments list in 2020. So looking at our research questions, going back to 2013 and 2014, I'm going to review with you some of the trends and study design from that that sample collection, and then go into how that compares to the new samples that we collected in 2021. The original study had set out to figure out which sources or host animals were contributing to the bacteria impairment in the river, which we realized is really boring to say, and basically changed the title to Who Pooped in the River. And so everybody laughs at that. And so we still put that in all our slides. But really, we designed that study initially with a lot of quality control in mind, having not done this type of study before, just knowing there is E. coli, but we don't have a clear idea of what the sources are. So that, are you sure that's who pooped in the river? That was our quality control element. We also, as part of that, included a very high sampling frequency. We were out sampling once or twice per week from April through October in both of those years, 2013 and 2014, in order to get a clearer idea of the spatial and temporal trends, and use that to determine whether any pollutant source types or locations would be highlighted by the data. Our follow-up study, we did a small one-day sampling in 2016, and then this larger study that was done this past summer and fall in 2021. That study was designed as to match the methodology from the original study as much as possible, with a general hypothesis that our human poop problem was still present in the river and we just needed to figure out where. So this follow-up study was designed with a higher sampling density and lower sampling frequency in order to zoom in on the potential spatial trends and hotspots that we might find along the river corridor, as well as addressing a data gap from the original study, which was the potential confounding factor of detecting already treated wastewater treatment plant outflows, basically treated human sewage where the cells could still be detected before that they degraded in the river, but would not be a human health risk. So this study included a piece to address that question. So just real briefly on fecal indicator bacteria, most of you are probably familiar with this, but in both the original 2013 and 14 study and our recent follow-up, we collected multiple samples to analyze host sources using both Bacteroides and E. coli to measure bacteria concentrations against state water quality standards for safe human contact. And so there aren't any current standards for Bacteroides, and so the E. coli was really important for that piece of the puzzle. And then in both studies, we used Bacteroides sample analysis. The microbial source tracking portion was Lumen Ultra Labs, formerly known as Source Molecular, and they're in Miami, Florida. And we had E. coli was analyzed at our local Farmington wastewater treatment plant lab. And both of these fecal indicator bacteria are not inherently dangerous in and of themselves, but they indicate that there is fecal material present in the water and could indicate the presence of more virulent pathogens. So why microbial source tracking? 10 years ago, when we were planning the original study, we legitimately did not know which of these suspects were potentially contributing the most to E. coli impairment. And so through a combination of stakeholder outreach, the work of the watershed group prior, working on source IDs and studies throughout the watershed in the earlier part of the 2000s, we worked with the stakeholder group and Source Molecular Labs to pick out the most likely suspects. And so in the original study, we tested all the samples for presence and absence of Bacteroides markers originating from human, cow, bird, horse, and dog. And the cow marker was eventually replaced with one for general ruminants. So you'll see ruminant replacing that later on. But as I mentioned before, in that original study, we had fewer sites and higher sampling frequency. And so we primarily looked at 4 sites: the Animas at Aztec, Animas at Boyd Park, the San Juan at Farmington upstream of the San Juan confluence, and then all the way downstream on the San Juan at Hogback, which is the border with the Navajo Nation. And then in 2014, we were able to add a 5th site at the state line to provide a little upstream background information, and that one had fewer samples, but is included in some of the results. Doesn't want to change. There we go. So looking at our percent positives from that first study, as you can see here in green, that's our human source bacteria. Human sources were present about 60% of the time at the 2 Animas sites and over 90% of the time at the 2 San Juan sites. The ruminant sources here in red, They were present about 90% of the time at all sites, and bird sources about 30 to 40% of the time at all sites. The ruminant and bird sources were kind of to be expected. We knew there's livestock sources out in the landscape. We know there's ducks and geese in the river. So those were kind of to be expected, but the human source bacteria was a big surprise, and the one that we thought was most possibly likely to cause illnesses downstream. And so that was the one that was focused on primarily as a follow-up after this study. And so we worked with Lumen Ultra to labs because of that increased interest in the human source bacteria. We worked with them to do a more detailed analysis of the actual quantifications, not just percent presence absence, but the actual concentrations of the human bacteria marker. And they used 3 different methods of varying sensitivity to calculate site averages for the Bacteroides dorii human fecal marker HF183. And so the 3 methods, as you can see, the columns for each site, they did not significantly differentiate the results within each site, but the results do highlight the stark increase in human fecal concentration between the Animas to the San Juan, and then the San Juan at Farmington downstream to the hogback, where the site average at that hogback site ended up being approximately 6 times greater than the illness benchmark of 4,200 copies per 100 milliliter, which was from a study in the literature of how the rate at which approximately 30 swimmers out of 1,000 could get gastrointestinal illnesses from the presence of bacteria. And so that was very concerning, and something that we definitely wanted to follow up on what is happening there that's causing those increases. This is just another way showing that same data, the box plots of the different sites. And you can see the hogback site again, the averages for human bacteria or the distribution of samples very high in the tens of thousands and hundreds of thousands in the high end of that range, and really just not in the range that we wanted to be seeing. This switches to showing the E. coli concentrations. It shows a similar trend where you're having increasing E. coli concentrations as you move downstream, with a lot of exceedances of our primary contact water quality criteria at those San Juan River sites. And that is one of the main reasons we focus solely on the San Juan River for this follow-up study. Now, the answers to our questions from the original study. So who pooped in the river? Ruminants, definitely 95 to 100% positive samples for that. Humans, 60 to 97% positive, depending on sites. Birds were 30 to 50% positive, and we had measured for horse and dog, and none of those had been detected. All of our quality control measures did lend themselves to confirming those results. We ended up doing 2 different human source markers as well as some backups, and so that really did, we felt very confident that those results were accurate. As far as the spatial magnitude, we saw a greater concentration of both the human B. dorii marker and E. coli in the San Juan River than the Animas, with the greatest increase being between Farmington and the hogback. And we saw lots of exceedances during monsoon season. So there was a positive correlation between E. coli and storm events, with increasing flow, turbidity, and nutrients during those time periods. And so this study also brought up a lot of additional questions. What are the actual sources? Do the data point to any geographic sources or rule them out? And could those wastewater treatment plants maybe be contributing, or could we just be detecting all of these dead cells continuing further downstream? And so these were all things that fed into the new study. We did have some small funding to do a targeted sampling in 2016. There was a known source, the Harper Valley wastewater treatment plant, that was being decommissioned in January of 2017. And so we ran out there as quick as we could and took some up and downstream samples. This shows, these red show the concentrations from that one-day sampling. So this was a couple sites in Aztec and Flora Vista. Upstream at Farmington, it was in the 10,000 range. And then here's right upstream and downstream of that Harper Valley plant in 2016. And we did see a jump in human bacteria concentrations right then, but they were already pretty high upstream of the wastewater treatment plant. And so that was just kind of a one-shot thing to look at a particular source. And that was kind of additional information to plan the sites for our next study. The way we did that was we really worked with the community to identify potential sources on the landscape. So are there areas that have high densities of septic tanks? Are there areas where there may be straight pipes or illegally permitted systems, things like that? And again, the wastewater treatment plant locations, could those be contributing? And so we worked with the community to identify sites and tried to bracket known or potential sources to see if any of those could be hotspots that are contributing. And so this is a map of the sites that we sampled in 2021, starting all the way upstream at the Bloomfield Bridge, sampling the Bloomfield wastewater treatment plant outflow on the main stem at McGee Park, downstream of Wild Horse Valley, which is a potential source at the start of the Bolac Ranch, the San Juan Farmington site, a repeat site from back in 2014. Same with the Animas Boyd Park. We wanted to make sure we're capturing our large tributaries as they came in. We did sample the Farmington wastewater treatment plant outflow, which is just downstream of the Animas confluence, about a quarter mile from this next site at the San Juan Vista Bridge. We sampled the La Plata River, a major tributary coming in, although we did not see very significant flows during any of our sampling events. And so that could have potentially confounded those results as opposed to during an event where the La Plata is flowing really strong. We sampled at those same two sites, San Juan Harper Valley, just upstream of that old wastewater treatment plant, and then Lions Park, which is the downstream site of that. San Juan Fruitland at the Fruitland Bridge there, the tributary with Stevens Arroyo, and then so a few sites downstream in water flow, and the Shumway Arroyo, with our final site being downstream at the Hogback on the San Juan. So the $40,000 question, this is our study budget. What were we gonna try to answer with this, this new study? So have those E. coli numbers and human source numbers changed since 2016? Our hypothesis was that we assumed that they were still there, that there were still human sources in the river, and that we were hopefully going to see where those are distributed along that set of sampling sites. So if humans did poop in the river, where, a little bit of when, we only had 4 sampling dates, so we had a lot less density to get into that temporal issue, but how much? And then are those wastewater treatment plants contributing? And if they're not, do any of the data point to any of the other potential hotspots? So I'm going to take a sip of water, leave you in suspense, and then we'll get to the graphs of the results. Okay, so these are our E. coli results. So these are very similar to the trends that we saw in 2014, with nearly all of the sites having at least one E. coli exceedance of the 410 CFU grab sample limit. We've got our E. coli on the y-axis here, and then our sample sites going from upstream to downstream here. I did realize with the San Juan flowing east to west, it's like a mirror image of my graphs. And so maybe we could just get the river to flow the other way, so Alyssa and I don't have to change the way the graphs are oriented, but just flip your imagination from the map. And remember, this is upstream and this is downstream. So yeah, we have exceedances of all of these, and we do have the wastewater treatment plant sites are on here with the O. And so you can see that all of these were 0, our 2 wastewater treatment plant sites. There was never any E. coli detections coming out of those outflows. But we did have exceedances, at least one exceedance at all of our other sites, with the exception of La Plata. So all of our tributaries and mainstream stem sites had at least one E. coli exceedance. And so what about the B. doria, the human source bacteria? And so this is the quantification graph for that is scaled for all of the results, including the wastewater treatment plants. And so back in 2014, we had our maximum ever detection for the human B. dorii marker was to 373,000, and that was at the Hogback site, and then 190,000 at the San Juan Farmington site, which is here. And so we did not reach any of those maximums. In fact, you can hardly see the results down here. The 2 wastewater treatment plant sites did have some high concentrations, but that did not seem to spike the sites that were further downstream. We'll show some of those data in more detail in a second. But as we zoom in on the scale here, These are the results showing, excluding those wastewater treatment plant sites. So just looking at the main stem and tributary sites. And you can see our scale went from 1.5 million down to 5,000. And so we only had one exceedance in the whole study of that illness benchmark, that 4,200. Whereas in 2014, we had many of the site averages were way above that. And so that's a huge improvement. And only 29% of the 2021 results were quantifiable for the human source bacteria, as opposed to in 2013 and 2014, we had the A. Boyd site, 61% of the samples were positive. In Farmington, 92% were positive, and San Juan Hogback, 97% were positive. So that is a major improvement. And so this is just jumping back to that previous result. So you can see these high numbers and how those concentrations, we did not see anything in that same ballpark in this past 2021 study. And so this is looking into detailed trends within each of our 4 sampling dates last year. You can see the yellow line here shows our E. coli concentrations, and that corresponds with the left-hand axis. And then our brown line is the B. dorii quantifications, and that corresponds to the right-hand axis, and that'll be the same for the next few graphs. And then the red line here is our NMED E. coli grab sample limit, so that compares to the yellow. So looking at the pictures here, you can see the river's pretty muddy. We did have a storm event on August 2nd that led to a 3,000 CFS spike on the San Juan River the evening of August 2nd. And in the original study, we found high correlations between E. coli and turbidity and storm events. And you can see here, we had all of our main stem sites were exceeding that grab sample limit on this first sampling date. And then we had the 2 tributary sites down here, Stevens and Chumley Arroyo, were also exceeding there. For the B. dorii, the majority of our samples were non-detects, with the exception of our 2 wastewater treatment plant samples, which you can see up here kind of floating off the axis. It is important to note that we had a shipping delay on the second day of sampling. And so we had, unfortunately, water samples that sat in the FedEx facility all weekend instead of getting in the lab in 24 hours. And so they were out of temperature when we reached the lab. And so that includes all of these sites. So it's potential that those non-detects could be based on the degradation of the bacteria over the weekend due to the high temperatures. But the lab basically said if there was low concentrations, it could have degraded to 0. If there had been really high concentrations, you would expect to see some still detectable. So we just needed to make that quality assurance note. We don't exactly know why those are non-detects, but as you'll see in the next few slides, those trends kind of did continue. Okay, so our next sampling date was August 25th and 26th. And as you can see from the pictures here, the river was a lot clearer. And we had a higher flow rate coming out of Navajo Dam, as well as being further removed from any storm events. And this does seem to be reflected in the lower E. coli numbers we are seeing. We only had 1 exceedance on the main stem and then 2 tributary exceedances on that sampling date. And then you can also see, despite these high wastewater treatment plant samples inflows, again, we had non-detects in the downstream samples. So those did not seem to be affecting the concentrations seen in the river, even at those lower flows. As we move downstream on the with the B. dorii, you do see a couple detections of the human bacteria as we get to Fruitland and downstream. And we did not see detections in the tributaries. So that did seem to be something that was added to the river within that stretch, and we're measuring that there. But again, these concentrations at 1,000 or less, still way lower than the 10,000, 50,000 plus concentrations we had seen pretty frequently in 2014. In September, it was another muddy day. We had some storm events again. There were spikes on both the 28th and the 29th of September that led to 1,500 and 1,900 CFS increases on the San Juan during our sampling. You can see on the yellow line that the E. coli exceedances start increasing as we go downstream with some very high concentrations on Stevens and Shumway Arroyo that could be driving some of those increases in the main stem further downstream. But 80% of the samples exceeded the E. coli limit on this day. And so it starts way upstream, where even at Wild Horse Valley downstream McGee Park, that reach is also exceeding that limit. But in contrast to the E. coli, you see pretty low B. dorii numbers all the way in this downstream reach, and just a handful, 2 exceedances in this upstream reach. And so kind of opposite pattern to what we'd seen on the first 2 sampling dates. And then finally, our October sampling was the last date where we were back to a state of a lower CFS release from Navajo Dam. And this was actually our lowest river levels of the study period. Alyssa was lucky enough to be on a raft one of these days and had to get out and push a few times. Her iPhone got sacrificed to the river gods in that exciting adventure. But yeah, this is our lowest flowish for sure. And the interesting thing, we initially did not have enough funding to collect any MST sampling on this day. It was just going to be E. coli. And we were able to scramble some funds together at the last minute and collect. We basically had funding for 7 sites, and we chose the 7 downstream main stem samples, and they all turned up positive on this one. So we're really glad we were able to get those funding together, because it's the kind of thing, if you don't measure, you never know what you're going to find. And so these were actually the highest B. dorii concentrations of any of the sampling dates, and including our only exceedance of that illness benchmark of 4,200. And so that was at the San Juan Fruitland site. And so you can kind of see these increasing a little bit and then dropping off, but still being positive further downstream. And interestingly, this was the only time in the study. So you see these low E. coli numbers and then a sudden bump of E. coli. And so this is really the only sampling point in the whole study where E. coli did seem to be correlated with the human source bacteria that could potentially human source could be driving those E. coli concentrations. They really seem to be pretty uncoupled other than this one point. So that's pretty anecdotal, but just sort of an interesting observation on that single sampling date. And so what are our conclusions from this? So have E. coli and human source back to B. dorii numbers changed since 2016? And the answer to that was a pretty clear yes. The E. coli numbers are still significant, but the human source levels are much lower than they were 8 years ago. And really, this was a big surprise to us. We were expecting to find all of these smoking guns, and we designed the study that way to hopefully find which tributaries were contributing and things like that. And that's not what we found. That's why you do the study. And so we'll go into that a little bit more in a second. But the other really important question was looking at those wastewater treatment plant outflows. Do we think that those are contributing human source levels to the river? And more specifically, could those have been what was driving the high concentrations that we measured back in 2013 and 2014? And so I think the answer to this is a pretty highly likely no. The high numbers were measured at the wastewater treatment plant outflows, but they did not seem to be driving increases in concentration further downstream, and likely do not represent a human health risk. We do know that with the methodology we use, the PCR used to do the microbial source tracking, they're looking at DNA markers. So is that DNA strand present? And so that can detect both living and dead cells, whereas the E. coli is a culture method where the E. coli have to grow overnight in an incubator, and so you're only detecting living cells. And so we think that most likely we're getting a high concentration of dead cells that are then quickly degrading as they move downstream outside of the wastewater treatment plant system. And this could be, there's methodology out there with the fluorescent DNA binders that this could be quantified more specifically in the future, but I don't really see that as a priority, but that could be an interesting future research question. So what about our spatial trends and how much, our magnitude of the problem? Our tributaries did not have any detectable B. dorii, except for, no, I think none of them did. Yeah, so no detectable Bacteroides in any of the tributaries, but they did have high E. coli, especially the Shumway and Stevens arroyos coming in on the downstream reach. We only had 29% of our samples were quantifiable for human sources, as compared to over 90% on the San Juan previously. And we had only one incident exceeding that health limit of 4,400. But contrasting to that, we still had almost half the samples exceeding the E. coli limit. And so despite the reduction in human source bacteria, we're still seeing E. coli impairment. There were multiple exceedances in both of the San Juan reaches, so from Farmington upstream as well as Farmington downstream to the hogback. And so those reaches both would requalify for being listed as impaired for bacteria for primary contact. We saw our highest concentrations of bacteria coming from those two arroyos. And so they're lower flow, but had high concentration. So those could be still contributing a significant load of bacteria to the San Juan River. And those were added as their own assessment units by NMED. And so this would confirm their recent impairment listing for E. coli. And so does our data point to or rule out any specific human source hotspots? The answer was really not consistent. And I mean, we designed the study with the assumption that our null hypothesis, we would have high human bacteria, and then we were going to narrow down where is it coming from. And so with the overall reduction, there wasn't really a consistent trend in where that bacteria is coming from. But we do still indicate with about just under a third of the samples still being positive, that there are sources that still exist between Bloomfield and Farmington and between Farmington and the hogback. And so as we continue to work to reduce human source pollution, on the ground projects and outreach should still focus watershed wide, since we're not exactly sure which specific improvements or methods led to these improvements. Focusing watershed wide, casting a wide net, hopefully will show us the best results long term. And then probably our most interesting result as far as planning, having that high E. coli but low prevalence of human sources probably means we do need to be focusing on bacteria from other hosts. So refocusing on that ruminant bacteria that was present so frequently in the original study, and working from there. And so the other big question that comes up from this is, what could have led to these decreases in human source bacteria? There were very significant infrastructure improvements between 2016 and 2021, the most recent sampling. The Farmington wastewater treatment plant underwent a large expansion, which included a sewer line extension to Kirtland that wrapped up the old Harper Valley wastewater treatment plant. That was a small operating wastewater treatment plant that was pretty much out of compliance with its discharge permits for years. It was on schedule to be decommissioned, but it just took a long time to get there. So that was a known point source that could have been contributing some of those very high downstream numbers. And so that and a next door sewage lagoon, those were both added to the Farmington sewer in, I believe, 2017 or 2018. There's also been a big effort since our study results were initially published in 2014, 2015, and on into the recent years. NMED and partners have focused on improving the certification process for septic haulers and pumpers, so that they need to take some classes, learn the procedures, make sure that things like illegal dumping and just improper handling of septic waste aren't happening. And so that level of education and certification of the professionals, as well as education of the community on best practices for septic care, all of those can improve and lead to benefits. So did we accomplish our mission? Can we take credit for this and pat ourselves on the back and retire? I don't know that that's necessarily true. The work concerning liquid waste has certainly been successful. It's had benefits to the river, but it definitely needs to continue. Since we don't have just exactly the clear before and after that this exact thing solved the problem, it's important to continue this work, especially if it's been the accumulation of individual actions throughout the watershed. And so the past 2 years, our funded grants through our Lower Animas watershed-based plan and some other funding sources have done a lot of outreach. And so we had 46,000 flyers go out to San Juan County utility customers in 2020 to make sure that they understand proper septic systems and how to care for those, how to report illegal dumping, things like that. Alyssa led a really great RV dump station sign improvement campaign last year. 16 roadway signs were installed around the county so that people who are coming as tourists to our area know how to properly help take care of their septic or their RV tank waste, and they're not accidentally contributing to water quality pollution problems just because of a lack of information. And so those signs are around the county, and then there's brochures with maps at a lot of gas stations and tourist destinations throughout the county. NMED worked on a liquid waste program rules and regulations training for septic professionals that was held at the end of last year. And Farmington has still a lot of sewer and infrastructure improvements on the docket. And so those big infrastructure projects cannot be underestimated. I think those were probably the #1 thing that has led to success as far as getting people, especially in some of the areas where septic tanks may have been installed before regulations, getting those people onto sewer and just having those options be available has been so important. And as we think big and into the future, I think there's a lot of opportunity for assistance to individual homeowners. A lot of times the people who are dealing with situations of failing septic tanks or surfacing sewage, things like that, it's not that they want to be polluting, it's that it's expensive, and it's kind of a economics issue and a really a social justice issue where people don't have the economic means to be doing what is best for them as a homeowner and best for the community and the watershed. And so if there are cost share programs available in the future to help people get over those economic barriers and remove a threat to them and to the watershed, I think those would be great examples of win-win solutions that could really be helpful into the future. And then as far as addressing our bacteria from non-human sources, that original study did detect those ruminant sources in really high concentrations. And we have been working really hard on the Animas watershed with doing, really, there's a lot of best management practices that help soil health, irrigation efficiency, pasture health, all of those kind of things, and also have positive benefits for reduction of bacteria runoff to the river. And so continuing these type of management practices all throughout the San Juan River, that's going to be a big benefit as far as reducing those downstream non-human bacteria sources. Alyssa has been working really hard on getting our draft San Juan restoration plan. That's been in the works for the past 2 years. We'll have a draft out by the end of this month, and that's going to hopefully lead into a larger scale watershed-based planning process where actually planning specific on the ground projects, calculating load reductions and things like that. But there's still a lot of win-win solutions out there, and hopefully those can move towards continued improvements to our bacteria. And with that, I can move on to questions. I saw some things popping up in the chat, so I don't know, Christine, or if you want to moderate questions, and we can go on from there. And I'll just
yes, that'd be
stop
great.
sharing.
Yeah, I was. Okay, yeah, we do have a lot of questions. So the first one is from Eric Rich, and it's, did you ever look at the Harper plant MPDES self-monitoring reports for E. coli discharges?
At some point we did. It's been a while. That was probably after our initial 2014 study ended. I remember downloading all those reports and also seeing that within NMED's regular sampling. That was one that was always kind of flagged as out of compliance. And so we do know that during our original sampling period was a time when that plant was out of compliance. And so I don't think we went into like the day by day to see if that would lead to any of the jumps we saw back then, but that would be really interesting to maybe look at in more detail while that was still in operation.
Yeah, sounds like the shutting it down and diverting made a big difference.
Definitely.
Okay, the next one is, what is the source of your illness rate benchmark?
That was from a report. Alyssa, do you remember where that was from? I know the lab Lumen Ultra had directed us to that. I think it was an EPA report.
Yeah, it was an independent report that was conducted from Stanford University, and that was the team had done a comparison of human source numbers directly taken from sewage plants, and they did several hundred repetitions of risk assessments specifically for human source bacteria. And after those several runs using previous data from previous studies, they came up with that 4,200 copies per 100 milliliter benchmark, which is not something that is a threshold that is used by the EPA and by NVD, but it's something that we're referencing for the sake of the study.
And that was one of the things we like when we did the original study, we were just floating in space with our data. We were like, is 100 too high? Is 1,000 too high? Is 10,000 too high? Like we really had no idea what we were referencing, because most of the studies previously that had been done were done on beaches in California. It was like where a lot, in Florida was where a lot of the things happened, where you had runoff coming in and they had to figure out whether to close the beach that day or not. And so that was what was driving a lot of those benchmarks. And so they would compare within their data sets to the other beaches, but there wasn't anything that was like combining literature of what to compare with. And so, yeah, back in 2014, we truly didn't know. Like we had 100 concentrations on the Animas pretty frequently, and we're like, is that too high? And so, yeah, it's really was kind of hard to know with these. The pro and the con of using these new technologies is that there's not as much in the literature to compare against. But we did finally find that benchmark, and that got us grounded a little bit.
If that's easy to pull up, would you mind putting that in the chat? I think there might be other people who'd be interested in it, or at least just how to reference it so folks can Google it.
Be happy to. We have a link to it in our microbial source tracking report. I'll put that.
Great, thank you, Alyssa. Okay, I have a question from Ben Martinez. What is the change in tube raft use on the San Juan Animas in that time frame?
That's a really good question. I don't have any data to specifically back that up. I do know that Farmington and San Juan County have been really trying to promote recreational use. And so I don't know if there's been any kind of like, I don't know, there was a lot of anecdotal evidence about potential illegal dumping of septic waste. And there were also some prominent cases of companies that got caught doing that and were punished fairly publicly. And so it's possible that if that was something that had been happening frequently before and then somebody got caught, that they just stopped doing it. Or potentially people see, oh, the river is actually an asset and not just a wastewater canal, and maybe we need to be taking care of it a little bit more. But I do know, like Desert River Guides was our partner on the raft sampling. They've started a raft company in Farmington doing economic development and recreation down there through Farmington stretches. And I know tube use has kind of always been popular. There's long stretches of only private land where there's no takeouts. And so I think that it's not as popular as say in Durango, where there's lots of public places to get out of the river. But it's definitely, in the summer, if there's water in the river, if there's enough water to play in, there'll be kids playing out there in Aztec and Farmington. And so, yeah, it's definitely important that we address these recreational uses.
Well, on that same note, Ben asked, did Farmington and San Juan County increase the number of toilets along the river for river uses?
I honestly don't know about that. I would have to defer to maybe Farmington Parks and Rec or something like that. Because yeah, outdoor defecation, that was one of the things where we had no way to calculate before or after what contribution that could have been coming from. Pretty much always when we brought up this issue, people would bring up the septic tanks. And so that was where we had really bracketed upstream and downstream of a few of these communities where we knew that there was a really high density of septic tanks that were all installed before the permitting regulations came into being. And there was basically within the floodplain, not exactly the right substrate, where if they were permitted today, they would have to be more advanced treatment or above ground systems. And so that was one of the things where we did not see any specific trends upstream and downstream of those communities. It doesn't necessarily rule it out. It could be based on the time of day, all sorts of different things, but there wasn't any smoking guns that appeared from any of those sources.
Okay, great. Eric has another question. Ruminant versus human sources of pharmaceuticals would be a wonderful study using this same approach.
Yeah, we did look into some of those other background. We had looked into, I think, caffeine, and we had originally done a fluorometry to detect optical brighteners, which can be a source of, those are often present in wastewater, because they're in all soaps and detergents and things like that. But yeah, the pharmaceuticals would be interesting because, yeah, cattle take antibiotics too sometimes. And so, yeah, that would be really interesting. But I've heard a lot of interesting things like caffeine, ibuprofen, different things that you can detect in the wastewater that is present if human waste is getting into there.
And I think we're done with the questions. We've got one comment from Ben is that there are large homeless camps on the river in Farmington.
That is correct. Yeah. And so I think that's something that the city has been working to address, but that is one thing that we did try to capture in some of our sampling sites. For better or for worse, some of them had been in the really dense Russian olive stands along the river, which the conservation district has also been working to take out. And so I know our contractors worked really closely with City of Farmington, police and fire departments to make sure that those camps were relocated before any of those removal projects happened. But I do know some of those really dense camps near the Bisti Bridge aren't there anymore after those dense stands of trees have been removed. And so I'm not exactly sure where that is, but that's obviously an example of where addressing a social issue and like a humanity issue would also have benefits to the watershed.
Yeah. Eric has another comment that Navajo Nation found possible ruminant sources of antibiotics in McElmo Creek.
Interesting. And that's another good point to bring up. So Alyssa and I have also been working with Lucy Parham at State of Utah and with the WIN Act group on another project to compile E. coli data throughout the entire watershed. That compilation is complete, and we're going to be passing off that data set to another contractor to complete the analysis. And so, but that would be a really interesting one. I don't know if that ruminant pharmaceutical stuff would have been included in bacteria data, but we'll try and at least make a note of that to make sure that that data source can be referenced as part of that larger compilation of data.
Great. Any other questions from folks who are here? I think it's an issue that affects almost all of us in our work.
I just want to say thanks for all those great questions. And we can definitely share the PowerPoint. And I believe Christine said this will also be recorded and available on their website.
Right.
And Alyssa has put together a final report that's going to be submitted to NMED for our the final deliverable on our 604B grant. And so I believe those reports will be public. Heidi, you can correct me if I'm wrong on that, but we should also have a written report that will go with this analysis.
Any other questions?
And Heidi says, yes, those are public. So we'll also have that report available once it's finalized.
Pretty impressive to see the change between 2013, 2014, and 2021. And
it was such a big change that we
was.
didn't know whether to celebrate or to like question what happened, because yeah, it was remarkable. We were very surprised.
Well, it shows that the good work you've been doing paid off. I mean, it sounds like it was public education as well as, the wastewater treatment plant shifts, and you got rid of one direct discharge of E. coli.
Yeah, the city investing in that infrastructure, like they should get major kudos for that.
Yeah, it looks like it made a huge difference. And really interesting that some of the work that we've been doing where we've got E. coli impairments, looking at dead, where it's downstream of a wastewater treatment plant, the dead versus alive cells. And I know that is more expensive. So if you're just doing presence absence, it's a little bit easier to say, oh, it's here or it's not here. Mm-hmm. But the way you structured the study was great, because then you could see that, well, downstream there wasn't a spike in E. coli. So you were just getting probably a lot of dead cells from the effluent. Any more questions? Terrific job, both of you, and great presentation. And wow, a lot of work went into this. Pretty exciting what you found during the study. And kudos to both of you for all the hard work that's paying off. So it's terrific. It's nice to see success as opposed to degradation. And so definitely something to celebrate.
I had to remind myself that I shouldn't be disappointed to see lower concentrations. I'm like, that's a good thing. But no, we appreciate all the support and interest, and yeah, happy to come talk about proof anytime.
Well, thanks to both of you for presenting today, and thank you to all of you who attended. This is our last Lunch and Learn for the spring, and we will be starting back up in September. If anybody has a presentation they'd like to make or suggestions, just shoot me an email and I'll start putting together the agenda for the scheduling the presentations for the upcoming Lunch and Learn in September. Hope you guys all have a great summer and a good field season. And congratulations to Melissa and Alyssa moving on to new challenges and adventures. And hope to see many of you back when we return in September. So have a great rest of your day and a great summer.
Thanks, everyone.
Thanks. Bye.